Everybody, I'm here with Dr. Jonathan McLatchie, and tonight we're going to be talking about intelligent design. We're going to be talking about the argument from design, particularly from biology. A lot of times these days, it seems like apologists would rather just talk about design through like maybe fine-tuning argument or different things like that. And it seems like the argument from like biology and other arguments from design seem to got a bad rap. They've taken a lot of negative heat over the last 20 years, and but Dr. Jonathan McLatchie is not one to be swayed or back down by the popularity kind of contests that are out there. And so, yeah, he's going to give us a presentation, the case for intelligent design. And I think that you're going to find that it's overwhelming. And so thank you so much for joining me, Dr. McLatchie.
Thanks so much, Eric, for having me on. It's an honor to be with you again.
Yeah, definitely. And so glad to have you back on. And this is a bit different. Normally we talk about historicity of the Gospels on this channel and things like that, the argument for miracles. But I think this is good. And so before we jump into the presentation, tell us a little bit about your background. I mean, you have a degree in biology and all kinds of education in this background. So it's not like you're just coming from this, without a lot of knowledge on this particular topic.
Sure. So I'm an assistant professor of biology at a small liberal arts college called Sattler College in Boston, Massachusetts. I'm also a fellow of the Discovery Institute Center for Science and Culture in Seattle, Washington. My PhD is in biology. I did my PhD dissertation on the eukaryotic cell cycle, which is a really fascinating piece of molecular machinery that allows our cells to divide. So that's my education. I also have 2 master's degrees, one in evolutionary biology and systematics from University of Glasgow, and one in medical and molecular bioscience from University of Newcastle. And I also have a bachelor's degree in forensic biology. I also run a ministry called talkaboutdoubts.com, where we basically offer private, free mentoring to Christians who are struggling with doubts about their faith. And we have assembled a team of more than 60 scholars and experts in different fields who are willing to do one-on-one private mentoring calls with Christians who are struggling with doubts, and indeed ex-Christians who want to explore whether there's a rational way back to faith. And basically the way it works is someone arrives on our website, submits a form, and then we distribute that to one of our volunteer experts who will then get in contact with the individual to set up and schedule a Zoom call to talk with them about their doubts and confidence. And so that's a little bit about myself.
Yeah, definitely. And guys, if you haven't taken advantage of Talk About Doubts and you do have things that are gnawing at you, that are bothering you, and you would like to talk to somebody, maybe you've talked to your pastors or somebody else and they just didn't quite have the answers for you, definitely recommend. I'm one of the counselors. There are a number of different experts, and it's just an amazing ministry. I'm really honored to be a part of it. And so I will definitely put a link in the description down below so you can take advantage of that. And so awesome. Well, Jonathan, I'm going to let you basically kind of take over here and just kind of run us through the positive case for design. And then at the end, I'll probably have some questions, some common objections to the design argument. And if you guys have questions in the chat, definitely ask those questions. If you want to do a super chat, those questions would be preferred, but we'll get through as many questions as we have time for. And so, yeah, let me get your slides on the screen here. There you go. So take it away.
Excellent. Well, thanks so much, Eric, for the introduction, and looking forward to presenting with you guys on the case for intelligent design, which is a topic about which I'm particularly passionate and fascinated. Let's just begin, though, by laying out a little bit of agenda for what we're going to be talking about this evening. So I'll start with laying out what actually is intelligent design. What do we mean by that? How do we go about detecting design in nature? And what is the evidential argument for the reality of design in nature? Then we'll look at the fitness of nature for the carbon-based cell. There's a lot that could be said on this. I'm just going to give you a small sample of some of that evidence. We'll look at the finely tuned genetic code, the evolvability of protein folds, irreducible complexity on the cellular level, the problem of reductive evolution, and then we'll draw our conclusions. Now, I'll say at the outset again that this is a whistle-stop tour of some of the arguments for intelligent design. Of course, there's a lot more that could be said and written and spoken about on this subject. There's a lot of books I'd be happy to recommend where you can go and get more. I could literally talk for hours on each of these individual topics, and there's a lot more topics that could be added here. So again, this is a very, very basic introduction to these topics. So let's begin with, what is intelligent design? So first of all, how do we go about detecting design in nature? Now, here on the screen, we see a cliff face, and most of us, I think, wouldn't immediately infer that this cliff face is the product of design. There's nothing about it that signifies that it was chiseled into the rock by some intelligent mind or agent. But when we go to this cliff face, this is a cliff face from Mount Rushmore in South Dakota, where you see the faces of 4 U.S. presidents, we immediately infer that, no, this isn't the product of wind and erosion. This is actually the product of a mind. Someone's actually taken it upon themselves to chisel into the rock these patterns that happen to conform to the faces of 4 U.S. presidents that we all know and recognize. Now, here's another example. Which of the following is designed? Well, both of them are equally complex. It's not that one is less probable than the other if you're just randomly punching keys on a keyboard, but we immediately infer the second one is designed by an intelligent agent. Although the first one, maybe we can't rule out that there was a mind involved in arranging those characters. There's no justification for inferring that such a mind was involved. And so when you hopefully see with the second example there, in the beginning was the word, that's a part of a verse from the first verse. That's the first verse of John's Gospel. And we not only recognize that it's complex, but it also has a semiotic dimension. It conveys meaning and functional specificity. And likewise with the cliff face that we looked at previously, it's not that the faces of those U.S. presidents that are carved into the rock is less probable than the random cliff face. Rather, the Mount Rushmore example has a semiotic dimension. It conforms to some independently specified pattern. And so we immediately infer this is a product of a mind, not unguided stochastic processes of chance and physical necessity. Now, how should we think about evidence? How should we think about what does it mean to say that X is evidence for Y? Well, I will own that I'm a card-carrying Bayesian, and the way that I would define evidence is that which makes a proposition more likely to be true than it was previously. And evidence for me is expressed as a likelihood ratio, that is the probability of that evidence existing given that hypothesis being true versus the probability of that same evidence existing given that the hypothesis is false. So to take an example, say that we have a court scene and the expert witness comes forward and he presents the murder weapon that was used to commit a crime, and he shows that the accused's fingerprints have actually been identified on the handle of the murder weapon. Well, that doesn't necessarily prove that the defendant is guilty, but it is evidence that tends to raise the probability of the hypothesis of guilt by virtue of the fact that it's more surprising that his fingerprints would be in the murder weapon were he not guilty than it would be if he were guilty. In other words, the evidence is more likely if he's guilty than if he were not guilty. And so it raises the probability of his guilt, and therefore it counts as some evidence in favor of that hypothesis. Now, it's not necessary to say, this is a very common misconception about evidence, it is not necessary to say that in order to count as evidence for a hypothesis, the hypothesis has to strongly predict the evidence in question. So an example that our mutual colleague, Dr. Timothy McGrew, likes to give is, imagine that you are out in the forest, far away from roads and civilizations and so forth, and you happen across this cabin. And at first blush, it looks as though there's no one inhabiting the cabin. And you decide, nonetheless, to inspect. And so you prise open the door. And lo and behold, you find a cup of Earl Grey tea that is sitting on the tabletop. And it's still steeping. It's not at room temperature. Now, would you have predicted with a high level of confidence on the hypothesis that the cabin is inhabited, that you would find such a cup of Earl Grey tea? Well, no, not particularly. In fact, it's probably quite unlikely that you would find make such an observation on the hypothesis of the cabin being inhabited. However, even though it's an unlikely observation, given that hypothesis, it's still far more probable than it would have been on the falsity of that hypothesis. And so that is the nature of evidence then. And so when it comes to inspecting, say, the information content of the cell and so forth, and these designoid features that we're going to go on to discuss, it's not necessary to show that intelligent design predicts these observations with a high level of probability. Rather, it's only necessary to show that it's far less surprising that you'd make those observations on the hypothesis of design than it would have been on the falsity of the design hypothesis. So it's not particularly surprising, for example, that you would find information content, language conventions, information processing, storage, retrieval apparatus in the cell on the supposition that a mind was involved, but it's wildly surprising on the falsity of that hypothesis. And so it tends to move the needle, I think actually overwhelmingly so, in the direction of design and by extension, theism. So let's move on then to our first argument that we're going to adduce for design. And this is an argument that I derive from a medical doctor and medical geneticist, Michael Denton, who has written a number of books called the Privileged Species series. And his most recent book is The Miracle of Man. Highly commend to your reading. This particular example I'm going to give, I derive from his book, The Miracle of the Cell. It's a really beautiful piece of teleology in nature. And again, I'm just giving you one example, but if you want, if you want to feel like a kid in a candy store, then I highly recommend checking out Denton's work. The Miracle of the Cell is a masterpiece in terms of some of the incredible prior fitness of nature that's necessary for life to exist, and certainly complex life to exist. So, the material substances of the cell are made up largely by they're made up entirely by the non-metal atoms: carbon, hydrogen, oxygen, and nitrogen. These make up, the material substances of the cell, and these are the only atoms that you could use to build a biochemical system. And there's a good reason for that. The reason for that is that they make strong, stable directional chemical bonds. And critically, these covalent bonds give molecules with shape. And it is shape that is the essence of biochemistry. These atoms have an electronegativity such that the attraction of electrons for hydrogen and carbon is very similar. And this means that you generate, by putting carbon and hydrogen together, nonpolar molecules. Carbon and hydrogen have a similar value for electronegativity, or how strongly atoms pull electrons towards the nucleus. And that means that they're able to share electrons, creating a covalent bond. And since carbon and hydrogen have very similar electronegativity, the electrons are shared equally, and the bond is nonpolar, which means that the electrons are being shared equally between the two atoms. But if you get, if you put oxygen and hydrogen together, then you get a polar molecule. And that means that there is not an equal distribution between um, in the sharing of those electrons between the atoms that contribute to that molecule. And this is critical to the whole organization of the cell because it gives you the hydrophobic force. And it's a hydrophobic force that organizes a higher structure of the biological realm, right? It's a hydrophobic force that assembles membranes and proteins that you see here. So this is a cell membrane, and you can see outside you've got water, and this membrane is known as a phospholipid bilayer, and the phospholipid bilayer has a hydrophilic component, which is facing the exterior, and therefore is making contact with the water, actually forms hydrogen bonds with water, whereas the hydrophobic component is buried inside the interior of the membrane. And so this is a phospholipid bilayer, and so this hydrophobic force is essential for membrane structure. It also plays a critical role in the formation of protein structures because the hydrophobic components are buried, they cluster together, they form aggregates within the interior structure of the protein, and so they're buried here in the middle, and the hydrophilic side chains of the amino acids are forming hydrogen bonds with water molecules, and so they face the exterior of the protein structure. And so the hydrophobic force is absolutely critical for really any form of life, because you need to be able to organize these biochemical structures. Charles Tanford, describing the discovery of how proteins fold, says, and I quote, "The hydrophobic force is the energetically dominant force for containment, adhesion, etc. in all life processes. This means that the entire nature of life as we know it is a slave to the hydrogen-bonded structure of liquid water." So here's the argument for design. The very atoms that give you stable, defined shapes from which you can build macromolecules also give you the hydrophobic force, which is the key to assembling them into higher three-dimensional forms. I find that to be absolutely mind-blowing and earth-shattering, that the very atoms that give you stable, defined shapes from which you can build macromolecules also happen to be the very atoms that give you the hydrophobic force, which is the key to assembling them into higher three-dimensional forms. That is a remarkable coincidence, and it's not the only example by any stretch of the imagination of these teleological coincidences that we find that form that give us the prior fitness of nature that's necessary for life to exist, and in particular complex advanced life. For more on this sort of teleological argument, I highly commend to your reading Michael Denton's series called the Privileged Species series. If you want the overview of all of these arguments, he gives kind of a summary of all the arguments and all the different categories. His book Miracle of Man, which is his most recent one, does a really good job of synthesizing his prior work. So let's move on then to another category of evidence for design, and this is the finely tuned genetic code. So the genetic code of life is the mapping that exists between codons and amino acids. So what are codons? So codons are essentially the words that make up the language of DNA, right? DNA is transcribed by an enzyme called RNA polymerase into an intermediary molecule called messenger RNA, and then it's taken out the nucleus and into the cytoplasm, where at the ribosome it gets translated into a protein, and the protein might form a part of a biochemical pathway, or it might contribute to some macromolecular machine or something like that. And the building blocks of these protein structures are amino acids, and each amino acid is specified by a triplet of nucleotide bases. Nucleotide bases form the building blocks of DNA, if you will. And so these three-letter words code for different amino acids. And the sequential arrangement of the amino acids in a protein will, by virtue of the properties of the side chains of those amino acids, determine how a protein will fold into its three-dimensional structure. And there are 64 different codons. There's 20 amino acids. So there's a so-called genetic code redundancy where amino acids are specified by multiple codons. So, for example, you can see that the amino acid serine is specified by UCU, UCC, UCA, UCG, and so on. Proline is specified by CCU, CCC, etc. You also have 3 stop codons, which are basically like periods or full stops in a language, and that signals for the ribosome to stop translating into a chain of amino acids. And what's remarkable is that there's actually multiple levels at which the genetic code is finely tuned, is highly optimized to minimize errors that might be incorporated during copying. So the genetic code, it turns out, that's found in nature is exquisitely tuned to protect the cell from the detrimental effects of substitution mutations. The system is in fact so brilliantly designed that codons, those 3-letter words, differing by only a single letter or single base, either specify the same amino acid or an amino acid that's a member of a related chemical group. In other words, the structure of the genetic code is set up to mitigate the effects of errors that might be incorporated during translation. And so that's remarkable. You change these letters by just one word, and because of the one letter, and because of the redundancy of the genetic code, you either get the same amino acid or an amino acid with similar chemical properties. So that's one level at which the genetic code is finely tuned or highly optimized for error minimization. Another level at which the genetic code is finely tuned are so-called encrypted stop signs. So another astonishing feature is that the sequences of stop codons overlap with those of the codons specifying the most abundant amino acids. So actually, I'm going to go back here and just show an example. So the most abundantly used amino acid is glutamine, which you see down here. And you can see that its codon GAG differs by only one letter from UAG. And this other codon that specified glutamine is GAA, differs by only one letter from UAA. And so, so that there's a reason for that. So this means that the genetic code is set up in such a way to dampen the impact of what are called frame shift mutations. Now, what's a frame shift mutation? Well, a frame shift mutation is where you have an insertion or deletion into the DNA sequence that's not divisible by 3. And because of the way that the genetic code is structured, where you have 3 letters for each word, you shift the whole reading frame. And so what this feature of the genetic code ensures is that when you have these frame shift mutations, it will generate stop codons. Because you have this overlap between the most abundantly used amino acids and the codons that specify them and the stop codons. And so that will stop the translation, and it will make sure that you don't get misfolded proteins as a result of the frame shift mutation, which would otherwise be quite catastrophic for that protein. Here's a paper. So this is widely established, by the way. This is not some fringe science I'm giving you. This is very well known, and there's a lot of literature published on this. So for example, there's this paper published in Nature. The genetic code is nearly optimal for allowing additional information within protein coding sequences. So they note that in the conventional genetic code, translation of a frameshift error is halted faster on average than in 99.3% of alternative codes. Now you might say, well, yeah, but there could be even more highly optimized genetic codes, and that is true for that particular one variable. But when you consider that there are actually multiple levels, multiple layers of fine-tuning of the genetic code, it's remarkable that so many different layers of fine-tuning of the genetic code also are close to the global optimum for fine-tuning or optimization. Related to this is the ability, also reported by the same paper, of the universal genetic code to, quote, allow arbitrary sequences of nucleotides within coding sequences much better than the vast majority of other possible genetic codes. And there was a paper on this topic that was published in the journal Biosystems titled Optimality in the Standard Genetic Code is Robust with Respect to Comparison code sets, and it talks about some of the fine-tuning features of the genetic code. There's also the choice of amino acids that are used, and so this is also something that's come to be quite well established. So this is a paper published in the Journal of Astrobiology. Did evolution select a non-random alphabet of amino acids? And the paper reads, and I quote, "The standard alphabet exhibits better coverage, i.e. greater breadth and greater evenness than any random set for each of size, charge, and hydrophobicity, and for all combinations thereof." In other words, within the boundaries of our assumptions, the full set of 20 genetically encoded amino acids matches one hypothesized adaptive criterion relative to anything that chance could have assembled from what was available prebiotically. Another paper on the same thing, titled "Extraordinarily Adaptive Properties of the Genetically Encoded Amino Acids," says, and I quote, "We defined an adaptive set of amino acids as one whose members thoroughly cover relevant physical chemical properties or chemistry space. Using this metric, we compared the encoded amino acid alphabet to random sets of amino acids. These random sets were drawn from a computationally generated compound library containing 1,913 alternative amino acids that lie within the molecular weight range of the encoded amino acids. Sets that cover chemistry space better than the genetically encoded alphabet are extremely rare and energetically costly. Further analysis of more adaptive sets reveals common features and anomalies, and we explore their implications for synthetic biology. We present these computations as evidence that the set of 20 amino acids found within the standard genetic code is the result of considerable natural selection. The amino acids used for constructing coded proteins may represent a largely global optimum, such that any aqueous biochemistry would use a very similar set." Now, this raises an interesting question, these different features of the fine-tuning of genetic code. And that's not even all of the fine-tuned features, by the way. This is a sample. An interesting question arises, though. Could you evolve a genetic code by natural selection, by some sort of trial and error process? The answer to that question is no. Changes in codon assignments would be catastrophic to the cell, because such a mutation will ultimately lead to changes to the amino acid sequence in every protein produced by the cell. I mean, think about it. If you're changing the codon to amino acid mapping system over time, every time you every polypeptide that's made by the cell is going to have different amino acids plugged in because you're changing the codon amino acid assignments, and that's going to wreak absolute havoc to your cell. So this means that one cannot have a significantly evolving genetic code. Yes, there are some minor variations to the genetic code. Mycoplasma, for example, has very minor variation to the genetic code. But these are very, very minor differences. You cannot really get a significantly evolving genetic code. This is why Francis Crick, the co-discoverer with James Watson of the double helical structure of DNA, described the genetic code as a frozen accident, because once it's established, it's very, very difficult to modify it over time. Now, some have tried to argue around this by postulating that the lesser used codons could be perhaps redesignated to a different but related amino acid, thus allowing the genetic code to become optimized. But there are, I think, significant difficulties with that proposal. For one thing, it seems highly unlikely that by virtue of replacing some of the lesser used amino acid assignments with a related amino acid, that one could attain the level of optimization which we find in the conventional code. And furthermore, the question is naturally raised, of course, as to what selective utility would be exhibited by the new amino acids, because they would have no utility until they're incorporated into proteins. But that won't happen until they're incorporated into the genetic code, right? And then, of course, let's not forget the necessity for the dedicated transfer RNAs, which are these adapter molecules that line up the amino acids. They have anticodons that are complementary in base pairing to the codons, and they basically line up the amino acids in the ribosome to be added to the growing polypeptide chain, and there are activating enzymes called amino acyltransferase synthetases, which are specific to the amino acids, and these are needed for them to have them in the code. And so there's really significant challenges, I think, to evolving a genetic code by natural selection. And of course, remember, the amino acyltransferase synthetases are themselves coded for by, or they're themselves made from amino acids, right? Because these are themselves proteins, and so they are made from amino acids. So there's a causal circularity problem there as well. So incredible evidence of complexity and design at every level here. Let's look at another argument for design, which is the evolvability of protein folds. So protein folds, proteins make up the enzymes that we find in cells that catalyze chemical reactions, speed them up by lowering the activation energy. We also proteins also contribute to macromolecular machines in the cell, or nanomachines, that govern the show in biology. And these are made up of protein components that work together in unison to achieve some higher level objective. And it turns out that even evolving single new protein structures is prohibitively difficult. As I mentioned previously, there are 20 different amino acids used in cellular proteins. And that means that for a modestly length protein of 150 amino acids, there are 20 raised to the power of 150 possible combinations. And so a question that we can ask is, how prevalent are functional protein folds in combinatorial space? Because if it turned out that the ratio is astronomically small, then it would be very difficult to envision how you could, by an unguided stochastic process, find those protein structures within that vast array of combinatorial possibilities within the time allowed in the history of life. On the other hand, if it turns out that they're relatively abundant, then it's much more likely that you would be able to do so by an unguided stochastic process. And there's been a number of papers published on this topic. The classic one is this one by Doug Axe in 2004, estimating the prevalence of protein sequences adopting functional enzyme folds. And he was basically doing a site-directed mutagenesis experiment on an enzyme called beta-lactamase, which is an enzyme that confers antibiotic resistance to some bacteria. And he gave an estimate of 1 in 10 to the 77 power of the ratio of functional to non-functional protein structures. So that's a very, very astronomically rare sequence. It actually produces a stable and functional protein structure. But it's not the only paper that's been published on this topic. So here's a paper more recently, a little more recently, 2006, in the Journal of Nature. Robustness epistasis link shapes the fitness landscape of randomly drifting protein. And in their experiment, where they were inducing mutations in a particular protein structure, after only a few random mutations under weak selection, according to their paper, around a third of subsequent changes to a protein completely disabled it. After several more mutations accumulate, the protein is inactivated by slightly under 2/3 of subsequent changes. And after random alteration of less than 10% of the protein's initial sequence, it becomes permanently non-functional. So the fitness approach is 0. And so we can try to work out the rarity by back solving, if you will. So the number of sequences that differ from the optimal one by a given number of amino acids increases almost exponentially with the number. And so a random search would find a barely functional sequence long before it found an optimized one. And so estimating the upper limit for the probability of a successful trial has to focus on that neighborhood of sequence space. So in the study, after 5 to 10 mutations, roughly 2 in 3 mutations inactivate a protein. And therefore, it follows that 1 in 3 amino acids each position on average would correspond to a functional sequence. The rarity would then be less than a third to the power of the sequence length. And so that estimate very closely correlates with the estimate that Axe provided in his 2004 paper in the Journal of Molecular Biology, that only 1 in 10 to 77 sequences corresponds to a functional fold or domain within the protein. And of course, the actual rarity would be quite a bit more extreme, since almost no sequences are functional after 10% of a protein randomly changes. So this presents us with a probabilistic resource problem. There are approximately 10 to the 80 elementary particles in the observable universe, and since the Big Bang, roughly 10 to the 16 seconds have elapsed. The time it takes light to traverse the smallest distance, so the Planck length of 10 to the minus 33 centimeters, is 10 to the minus 43 seconds. And so multiplying together the 3 relevant factors, we can determine the maximum number of events that could have taken place since the observable universe, since its origin is 10 raised to 139th power. And so you see that we very quickly exhaust the available probabilistic resources. And so it presents us with a needle in a haystack problem. Locating the functional sequences within the vast sea of combinatorial possibilities by a random search is like finding a tiny needle in an enormous haystack within a very limited time. In the English language, there are far more ways of writing gibberish than there are ways of writing meaningful sentences and paragraphs and so forth. And it turns out the same thing appears to be true in biology as well. And so that's why by a random search, it's very unlikely that you're going to be able to produce a lot of meaning, given that there's infinitely more ways of writing of non-meaning of having non-meaning than there are ways of having meaning and functional specificity. And so I think this is a real challenge to Darwinian evolutionary biology. The problem extends far deeper, though, because when molecular machines require multiple cooperating proteins in order to perform their function, the probabilities multiply exponentially. And so this brings me then to the challenge of irreducible complexity. And the idea of irreducible complexity really was put forward by Charles Darwin himself in 1859 in the publication of The Origin of Species, where he noted that if it could be demonstrated that any complex organ existed which could not possibly have been formed by numerous successive slight modifications, my theory would absolutely break down. And so Darwin put forward a test by which he could potentially falsify his theory of evolution by natural selection. Of course, Darwin was completely oblivious to the complexity and engineering and information processing systems that we find in the cell. It was completely unknown in Darwin's day. In fact, Thomas Henry Huxley thought of the cell as a homogeneous blob of protoplasm, which we now, of course, know is complete rubbish. So Darwin couldn't possibly have known what we now understand about the complexity of life and its engineering prowess. So anyway, in 1996, Michael Behe of Lehigh University sought to take up Darwin's challenge. And he coined this term, irreducible complexity, and he defined it as follows. He says, by irreducible complex, I mean a single system composed of several well-matched interacting parts that contribute to the basic function, wherein the removal of any one of the parts causes the system to effectively cease functioning. And that's a quote from his book, Darwin's Black Box, published in 1996. And he gave as an example from everyday usage a mousetrap. And we're all familiar with mousetraps. They have different parts that work together to achieve a higher level objective. So they have a platform, they have a spring, they have a hammer, a hold down bar, a catch, they have staples to hold the trap together. And of course, the trap still might still not work if the holding bar is too short or the spring too lightweight. So the pieces have to be well matched one to the other to work together in unison to achieve some higher level goal. In this case, catching mice. And the idea then is, how do you, by an unguided process, without knowing what the target is, bring everything together needed to realize a complex end goal? And so this is why irreducible complexity, I think, points powerfully to design, because explaining the origins of irreducibly complex features requires a cause that possesses foresight. And only intelligence can visualize a complex higher level objective and bring together everything that's necessary to actualize that end goal. So William Paley, and it's one of Eric's heroes, and as he is mine, he famously in his book Natural Theology, he says, in crossing a heath, suppose I pitched my foot against a stone and were asked how the stone came to be there. I might possibly answer that for anything I knew to the contrary, it had lain there forever. Nor would it perhaps be very easy to show the absurdity of this answer. But suppose I had found a watch upon the ground, and it should be inquired how the watch happened to be in that place. I should hardly think of the answer I had given before given, that for anything I knew, the watch might have always been there. There must have existed at some time and at some place or other, or an artificer or artificers who formed the watch for the purpose which we find it actually to answer, who comprehended its construction and designed its use. Every indication of contrivance, every manifestation of design which existed in the watch exists in the works of nature, with the difference on the side of nature of being greater or more, and that in a degree which exceeds all computation. And so I think that Paley would have felt very vindicated by our modern advances in the life sciences. And I want to give an example. I'm going to give two examples of systems that I consider to be icons of irreducible complexity. The first of those is the DNA replication machinery. I'm going to show a short video. It's only a minute long. I'm not 100% sure if the audio is going to work, but if it doesn't, then that's okay. I just want to give you a sense of the complexity and the engineering prowess of this system. I'll talk a little bit after the video about some of its about some of its functioning. So if the audio doesn't come through, then just enjoy the illustration of how this system works to replicate and duplicate DNA in preparation for cell division.
DNA replication is carried out by a molecular machine called a replicator that pulls apart the double helix and makes an exact copy of each strand. In this visualization, we're going to examine the mechanism for copying DNA. The enzyme DNA polymerase synthesizes DNA molecules using the original molecule as a template for the production of a complementary strand. The genetic code in the template strand is paired to free-floating bases, matching cytosine to guanine and adenine to thymine. DNA polymerase creates an exact copy of your DNA code, making less than one mistake in a billion bases.
So as I said, Paley would feel very vindicated by what we now know about the engineering prowess and complexity and designoid characteristics of living organisms. So as I said, the purpose of DNA replication is to split is for the cell to for the cells to split into daughter cells. Each cell needs a copy of the genetic material, which is the DNA. And so DNA is copied in what's called a semi-conservative fashion, where each strand serves as a template for the synthesis of a new complementary strand. So in the two daughter molecules, each of these daughter molecules is going to have one old strand from the parent and one new strand. And this is achieved by what's called complementary base pairing, where G always pairs with C and A always pairs with T. And so that provides a copying mechanism for DNA. So here's just a diagram that shows how DNA synthesis is carried out. So DNA synthesis is carried out by a group of proteins that act together as a replication machine. So we have various proteins involved. You have there are initiation proteins that initially unwind the DNA double helix, prepare for copying. There's a helicase enzyme that you see here, which basically breaks the hydrogen bonds that hold the two strands of DNA together to unzip the DNA double helix. You have the single-stranded binding proteins, which you see here, which basically bind to the single strands of DNA to prevent them from re-annealing, binding together again during copying, because you don't want it re-annealing while you're in the process of copying. So these single-stranded binding proteins make sure that doesn't happen. You also have these DNA polymerase that actually performs the copying. You have a sliding clamp that clamps the polymerase onto the single strand of DNA to prevent it from falling off. You have a clamp loader that loads on the sliding clamp. You have a protein, an enzyme called primase, which synthesizes a short sequence of RNA at the beginning of replication, because the DNA polymerase isn't able to replicate without the presence of a 3' hydroxyl group. It's a chemical group that needs to continue replicating. So the primase has to supply that, and so that needs to that's needed in order for replication to begin. You also have enzymes called topoisomerases, which basically alleviates supercoiling ahead of the replication fork. Because of the torsional stress on the DNA double helix, it can result in supercoiling. Think about an old telephone cord supercoiling, and that can result in the DNA breaking if it's not fixed. And so there are topoisomerases that basically will cut one strand of DNA, pass the other strand through the break, and then reseal the break. It's an incredible process. Because DNA is anti-parallel, so the two strands run in opposite directions from each other. One strand has to be copied backwards, and so one strand actually gets looped around, as you might have seen in the video, and has to be synthesized discontinuously. So there are short RNA primers that are synthesized, and then short fragments of DNA known as Okazaki fragments that are synthesized from those primers, and then the primers have to get removed and replaced by DNA. The Okazaki fragments have to get glued together, and there's an enzyme called ligase that does that. And if any of those parts were missing, you wouldn't have a system that worked half as efficiently as it used to, or quarters as efficiently as it used to, but it would be broken. So, for example, if you were to knock out the DNA polymerase, no replication would take place at all. The RNA primers would be laid down at the origin of replication, but you have no further replication. If DNA ligase links the DNA fragments that are produced in the lagging strand, in the absence of ligase, the newly replicated DNA strands would remain as fragments. Without the sliding clamp, the DNA polymerase would frequently fall off the DNA template. It would be too slow for efficient replication. In the absence of RNA excision enzymes, the RNA fragments will remain covalently attached to the newly replicated DNA fragments. Without the DNA helicase, the DNA polymerase will stall because it cannot separate the strands of the template DNA ahead of it. Little or no new DNA will be synthesized. In the absence of primase, RNA primers cannot begin on either the leading or the lagging strand. DNA replication, therefore, cannot begin. Without the single-stranded binding proteins, the DNA strands would re-anneal before they could be copied. Without the topoisomerase enzymes, the DNA would supercoil and eventually break. And so, as I said, if any of those parts are missing, even one of them, you don't have a system that works half as efficiently as it used to, or quarters as efficiently as it used to, but it's a broken system. And so, how by an unguided process, without knowing what the target is, are you supposed to bring about such a system by chance and physical necessity, by trial and error? And I actually picked this particular example. I cherry-picked it because this replication, self-replicatability, is itself a prerequisite for natural selection, because natural selection requires differential survival, which in turn requires as a precondition that you have self-replicatability. So you can hardly invoke natural selection to explain self-replicatability without assuming the existence of the very thing you're trying to explain. Another example, which also is essential for self-replicatability of cells, is the bacterial divisome. So this is actually the equipment that's necessary for the bacterial cell to divide and differentiate into two daughter cells. And I won't go into all of the details on that because it would take quite a while, but I do want to hone in on one particular point, and that is the cell wall growth as the bacterial cell prepares for division. Because bacterial cell division, by a process known as binary fission, requires the growth of the cell wall as the bacterial cell elongates. And the process occurs by the severing of the cell wall and the synthesis of new cell wall material. There are enzymes that are known as autolysins that hydrolyze the beta-1,4 glycosidic bonds that link the cell wall building blocks, and the gaps are filled in with additional cell wall material. And since these enzymes can result in programmed cell death, they have to be very carefully controlled and regulated. And there's just this is just a diagram that shows the role of what are known as penicillin binding proteins in cross-linking the peptidoglycan precursors into the cell wall. And these proteins, these enzymes, as their name implies, called penicillin binding proteins, they're actually the target for penicillin antibiotics, because penicillin disrupts this cross-linking process and stops this cross-linking from taking place. And so basically, as the cell elongates, the cell wall is being severed by the autolysins, but it's not being actively rebuilt. And there's also another another protein called bactoprenol, which carries the precursors of peptidoglycan across the cell membrane. So as I said, the penicillin is the target for these penicillin targets these penicillin binding proteins and disrupts their cross-linking of the cell wall as the bacterial cell is dividing, and that results, because of the osmotic pressure, that results in the cell bursting, and so it's dead. Now, this presents us with an evolutionary enigma. Consider the following two observations. Critical to the elongation process is the severing of the peptidoglycan cell wall by the autolysins. But critical to cell viability is the resynthesis of the peptidoglycan cell wall. Breaking the cell wall can serve no adaptive utility until a mechanism has arisen for the simultaneous integration of peptidoglycan precursors. Indeed, that latter mechanism, the cell is rendered non-viable, right? And so, how do you by natural selection, select for the process of severing the cell wall with the autolysin enzymes and so forth, without having a simultaneous mechanism already in place to rebuild the cell wall. Otherwise, it's going to result in cell lysis, or the cells bursting because of the osmotic pressure if you cannot rebuild your cell wall. And we know how critical these penicillin binding proteins are precisely because they're the target of the penicillin antibiotic. So this, I think, is a really critical challenge to the evolutionary scenario again. And again, I chose this example because it's another example, in addition to DNA replication, of what I call irreducible complexity on steroids, because you cannot invoke natural selection until you already have differential survival, and you can't have differential survival without self-replicatability. So again, you can't invoke natural selection without assuming the existence of the very thing you're trying to explain. So finally, then, let's look at the problem of reductive evolution. So the Darwinian model of evolution holds that one of the key mechanisms of evolutionary innovation is the duplication of genes and the subsequent divergence of one of the duplicate copies to undertake a new functional role. So because the probability of a single gene stumbling upon a significantly different, yet functionally advantageous sequence is so small, the idea is that following a duplication of a gene, one copy is able to retain the original function, while the other is free to explore the vastity of combinatorial possibilities in search of some novel function, right? And it's widely believed that a duplicate gene has no phenotypic cost. So the phenotype is just the physical characteristics of the organism. So it doesn't have so it's widely believed that the duplicate gene has no disadvantage to the organism. And so that is to say, it's selectively neutral, neither here nor there in terms of the organism's fitness. And in such a state, it's thought that the gene is free to mutate, independent of selection constraints or pressure. When previously protein coding gene incurs the leverage mutations such that it no longer codes for a useful polypeptide, the gene is considered to be a pseudo gene, or a broken gene. Now, it turns out that there's this problem of reductive evolution. Essentially, let me back up a little bit for a moment. So most mutations, about 75% of all mutations, are selectively neutral, being neither here nor there in terms of the organism's fitness. Of the remaining 25%, the vast majority of them are harmful mutations. And this is very well known. There's a small minority of mutations that are beneficial. So you've heard of antibiotic resistance, insecticide resistance, ability of E. coli to grow on citrate, or whatever it happens to be. But of those beneficial mutations, which are in a very small minority, the ones that natural selection can act upon, the vast majority of those beneficial mutations, the vast majority of those beneficial mutations are loss of function, not gain of function mutations. And so the question is, okay, how are we supposed to rely on this mechanism to account for the complexity and design characteristics of life? So here's a paper that was published in 2010 by Angager et al., this team demonstrated that the process of reductive evolution was sufficient to prevent lines of E. coli from taking a simple two-step pathway to a new fitness function. So basically what they did was they had various lines of E. coli, and they incurred two mutations on a particular gene called TRPA, which codes for an enzyme that's involved in the biosynthetic pathway of the amino acid tryptophan. The first mutation partially inactivates the gene. The second mutation fully inactivates the gene. And they put these lines of E. coli in a medium that was deficient in the amino acid tryptophan. And you would think that there would be a very easily, classic Darwinian pathway from no function to partial function to full function. Now, guess how many lines of E. coli took that very easy adaptive pathway from no function to partial function to full function? None. In fact, what they did instead is they incurred further mutations to further break the gene, because it turns out there's an energy cost in expressing a broken gene. So So the authors concluded that the cost of transcribing a broken gene incurs significant fitness costs, and thus its removal is facilitated by positive selection. There's always going to be far more ways of gaining an advantage by breaking something than there are ways of gaining an advantage by building something. And so natural selection is always going to find the ways of getting an advantage by building things before it finds ways of getting an advantage by is going to find ways of getting an advantage by breaking stuff before it finds a way of getting an advantage by building things. Okay, so this was confirmed by a paper the same year in PLOS Genetics, which came to a similar conclusion in Salmonella. The researchers examined the genomes of Salmonella species, arguing that the evolution of bacterial pseudogenes is not an entirely neutral process. To the contrary, bacterial pseudogenes are often actively degraded by the forces of selection, and degradation of genes which do not code for proteins that perform regulatory functions is most probably due to the energetic cost of transcribing them into messenger RNA. Finally, I'll give this example, which also illustrates this point. So this was a decades-long experiment, one of the longest-standing evolutionary experiments in human history. In fact, the longest, not just one of the longest, the longest. This is by Richard Lenski at Michigan State University. And he basically he worked with E. coli, and he ran this long-term laboratory evolution experiment over many years, decades, and he observed what evolutionary adaptations were brought about in his E. coli. And one morning, after more than 30,000 generations, he found that a flask containing one of the strains appeared particularly cloudy, which suggested that there were more bacteria present than usual. And they discovered, in fact, that E. coli cells had developed the ability to eat citrate in the presence of oxygen. And so this was hailed by the popular press and media as an example of evolution caught in the act. This is an example of evolution generating a new function for an organism. Now, what's critical here is that, in fact, E. coli can already eat citrate when there's no oxygen. So in an oxygen-deficient environment, E. coli is able to grow on citrate. And in fact, subsequent work found out what was the molecular basis for that ability. So a protein that's able to import citrate into the cell has a control region next to its gene that turns it off in the presence of oxygen. And a mutation duplicated part of the bacterial DNA, and that placed a different control region from a nearby gene next to that importer gene, and that allowed it to work in the presence of oxygen. Now, there was a subsequent study called titled "Fine-Tuning Citrate Synthase Flux Potentiates and Refines Metabolic Innovation in the Lenski Evolution Experiment," and it turns out that there were that in order to optimize the cell's metabolism for citrate, there were a lot of loss-of-function mutations that finally tuned that metabolism. And so even prior to that critical mutation taking place, there was another mutation in a gene for a protein that makes citrate in E. coli that degraded the protein's ability to bind to NADH, which usually helps regulate its activity. There was another mutation to the same gene later that decreases activity 90%. There were the scientists used computer analysis, which indicated that the citrate mutation would be the citrate mutant would be even more efficient if two other metabolic pathways that were normally switched off were in fact turned on. And in fact, sure enough, they found that two regulatory proteins that normally suppress those pathways had been degraded by point mutations. And so, and they say in the paper, when citrate is a sole carbon source, computer analysis predicts optimal growth when there is no flux through the enzyme. In fact, any of that enzyme activity is detrimental. And so it makes sense to get rid of it, to incur mutations, to lose that those regulatory proteins. So in conclusion then, intelligent design is based on standard principles of scientific reasoning about the past, based on the historical abductive method. Intelligent design challenges the concept of unguided chemical and biological evolution. And the design inference is based on cutting-edge advances in molecular biology, which reveals the complexity, elegance, and engineering prowess of cellular machines. And I'll finish with that. Happy to take any questions or discussion. Thanks so much for your attention.
Yeah, thanks. That was awesome. And so Chances are maybe not everybody caught all of that because that was just a lot of really good information. So definitely go back, watch this stream again. That's kind of the nice thing about having these things there is you could always go over it. And as Dr. McLatchie said, this is just kind of the tip of the iceberg. There's so much more that he could say. And so for questions, I do have one from Wesley Basner here. He says, what do you think of John Sanford's work on genetic entropy? Are you familiar with that?
I am aware of the book title. I haven't read it, so I wouldn't want to comment on that.
Okay, no problem. This comes from Hurricane Havoc. What is the evolutionist's best rescue device for the ERV regulation of the placenta?
Yeah, so what he's talking about here is there are sequences called endogenous retroviruses, which is abbreviated ERV. And these are widely thought to be relics of past retroviral infections that have infected the germline of primates. Primates have been on the scene for about 55 million years and have undergone many rounds of infections by these retroviruses. And our genomes are full of these retroviral-like sequences. There are literally hundreds of thousands of them in our genomes. And so our genomes are thought widely to be something of a graveyard of these prior retroviral infections, which are no longer infectious agents, but they've now integrated and been into our genome and have been passed down through our germline from one generation to the next. And it has been widely thought for many years that these retroviral sequences are non-functional, that they serve no function within the host organism. This is now known to be false. There's a wealth of literature on this topic. So the example that this particular inquirer has in mind is so the syncytin proteins that form the syncytiotrophoblast, which is essential for the development of the placenta. Basically, they form tethers that holds the placenta together. These syncytin proteins are coded for by what's known as the ENV gene, or the envelope gene, of these retroviral-like sequences. And this has been found not just in humans, but other mammals as well. And it seems that these sequences are actually not homologous, so it actually happened more than one occasion. And so I think what he's asking is, how does, from an evolutionary perspective, how does one harmonize the fact that these retroviral sequences are absolutely critical for placental formation with the evolutionary model? I actually do think this is a good question, and I do think anyone really has a good answer to this question. I mean, one might postulate some sort of non-orthologous gene displacement hypothesis and say that, okay, whatever was previously coding for those syncytin proteins got replaced by a retroviral sequence. But that's a word salad. I mean, is that really a plausible type of mechanism? So yeah, I think it's an interesting question. I don't think anyone really has a good answer to that.
Okay. Thanks for your question there, Hurricane Havoc. And here is a super chat. Jose Martinez, thank you so much. What are your thoughts on process structuralism?
So I'm not super familiar with the literature on that topic. So yeah, so I don't like to comment on things that I don't feel like I'm an expert on. So I'll not comment on that one.
No, that's fine. You just have to make something up. I'm just kidding. Just kidding. That is what we don't want to do. While we're keeping the comedy here, 1 second. There was one that I thought was really funny. Where was it? Sorry, guys. Yeah, this was my favorite. I dare Jonathan to present this material to Aaron Ra. So,
so Aaron Ra is frankly a crank on essentially any subject. I mean, there are certainly a lot of critics of intelligent design who are much more sophisticated than Aaron Ra, but I don't think that Aaron Ra is really the person I would want representing me if I were on the evolutionary side of things.
Gotcha. Okay. Yeah, I kind of commented back. Yeah, I don't think Jonathan's necessarily shaking in his boots over Aaron Ra. So, um, here is Truth is Beautiful. Does intelligent design commit begging the question, do we have to know that God exists before we can infer a designer from design? Um, Seems to be a little bit backwards. How would you
No.
answer that?
So yeah, you're right. This is a little bit backwards. So this is a common misconception that often comes up, not just on this topic. I've seen it come up when discussing things like the resurrection as well. That you have to have independent evidence before we can postulate that hypothesis as the best explanation or is evidentially supported by the observations that you're inducing. And I think this is simply false. And in fact, it's methodologically problematic because you never have the first piece of evidence because you'd always need some independent evidence in order to have the first piece of evidence. So
Right.
no, I think that as I said before, I define evidence as a Bayesian in terms of a likelihood ratio, the probability of the evidence existing given the hypothesis is true against the probability of that same evidence existing given the hypothesis is false. And so to the extent that these observations are more expected on design than they would have been otherwise, they tend to favor the design hypothesis. And notice that it's not necessary for me to show that each of those arguments individually is sufficient to conclude design. I only need to show that they cumulatively justify and establish a design hypothesis. So each of the arguments individually might somewhat raise the design thesis, the probability of the design thesis, without necessarily justifying it. But when you take all the evidences together, in my judgment, it puts a design hypothesis essentially beyond question, especially when you take into account the physical science arguments as well. One other point I'd make regarding the prior probability or the intrinsic probability of design, I think that the arguments from the prior fitness of nature that I introduced from Michael Denton, I gave an example of that. There's a lot more to be said there. I think that does very significantly increase the intrinsic or the prior probability of design, because it suggests independently that there is design in the world and that God is preparing nature and its properties for the design and organization of life.
Right. And again, I mean, you gave several lines of different evidence here. And so what somebody would have to do, the critic would have to do, is provide an alternative explanation that would cover all of this data. And you covered a lot of different things. And so I think that would be quite difficult. I'm sure people have maybe given it their shot on some of these things, but some of these things were even new to me. And so not that I've done a real deep dive into this particular topic, but I mean, I noticed even with like irreducible complexity, the examples that you brought up are not examples that I normally would hear with irreducible complexity. And so that was interesting. One more question from the audience, unless people got more, and then I might grill you with a few questions here. What books should I read for apologetics starters? I recently bought my first one called Reason for God. And so maybe give some recommendations as far as like books on intelligent design. I know you mentioned Michael Denton, and then just apologetics kind of in general that you would recommend.
Yeah, so on intelligent design, I definitely recommend all of Michael Denton's books. The Miracle of Man is a good book for giving yourself an overview of the different categories of evidence he would deduce. And if you want to do a deep dive in each one, then read his other books as well. I'd recommend all of Stephen Meyer's books, so Signature in the Cell, Darwin's Doubt, and his most recent one, which is Return of the God Hypothesis. I'd also recommend Doug Axe's book, Undeniable, How Biology Confirms Our Intuition That Life is Designed, which is excellent introduction to this topic. I'd recommend, probably these would be the ones I'd recommend for starters on design. It's probably enough to get you started. In terms of apologetics more broadly, Eric and I both spend a lot of time in New Testament scholarship, and I'd recommend all of Lydia McGrew's books, obviously. Recommend Hidden in Plain View. I recommend The Mirror or the Mask, and her most recent book, The Eye of the Beholder. Now, she's also coming out with a new book next year, which I read recently, which is not yet published, but it's absolutely fantastic, which is called Testimonies to the Truth. And I'm really excited for that book to come out. I'm going to be giving that out to people who are interested in this topic, because it's a really excellent introduction to New Testament scholarship and the vast array of evidences and different categories of evidence for the reliability of the Gospels in Acts. I'd also recommend Peter J. Williams' book, which is Can We Trust the Gospels? Colin Heemer's book, The Book of Acts and Synoptic History. Craig Keener's work on the Book of Acts as well is excellent. I recommend Richard Baucham's book for the most part, which is Jesus and the Eyewitnesses, the Gospels as Eyewitness Testimony. So yeah, I mean, it's going to depend obviously on the category. I also recommend Titus Kennedy's work. So he has 2 books out, one on the archaeology of the Bible, which focuses primarily on the Old Testament, also has some New Testament stuff. And then there's his other book, which is on just the archaeology of the New Testament. Is a really excellent piece of work. So these are just some of the books I'd recommend for starters.
Awesome. Yeah, no, that's a good answer. And I would definitely second most of those. I've read most of those. I haven't read Titus Kennedy's book yet. I do have one of his, Unearthing the Bible. I just haven't got around to reading it yet. But and so I have another super chat here from Demand. Who's Demand? Do you see ID becoming more popular among science in the next few decades? Are they opening up to considering it as credible any more than in the past? And that was kind of ties into one of the questions that I wanted to ask you was, why do you think so many like intelligent people reject the argument from biological design? And that would even include many Christians. I think of like the people like Francis Collins and a lot of the people that write for like BioLogos and different things like that endorse theistic evolution. And so, what is your take on that?
I see intelligent design becoming much more popular. I think there's a lot more support for intelligent design than people realize. I think, unfortunately, there's a social pressure to not be public about one's intelligent design views. You might have seen Expelled, which documents many people losing their jobs or being denied tenure, denied research grants, etc., for coming out and saying, I think there's something to this argument for design. So it's not like there's freedom of inquiry when it comes to this topic, unfortunately. But I encounter more and more scientists who are sympathetic to or outright supporting of intelligent design, even if they're not willing to say so publicly. And I think there's a lot of more support behind the scenes than people realize. And I think they'll get to a point where there's a tipping point where we reach a critical mass, and then people are going to feel a lot more comfortable coming out and saying, hey, I think there's actually something to this argument. And at that point, the dikes will break.
Right. It seems like the more scientific knowledge that we gain, it seems to me like it gets more traction. And so, at least from my vantage point. Good question there. Thank you, Demand, for the super chat. Another one from Truth is Beautiful. Do we know enough about DNA replication when people object God of the gaps? And that was actually another question that I had written down. The God of the gaps argument constantly comes up when it comes to really any theistic argument, including intelligent design. And so how do you respond to the God of the gaps argument?
Yeah, so the God of the gaps argument basically says that we can't explain feature X, and so we just plug in God or designer as a placeholder for that of which we're ignorant. And as science advances and we discover naturalistic explanations of these phenomena, the argument has to retreat with each new advance in scientific learning. And I think that this argument is not a God of the gaps argument. Whatever else might be wrong with the argument, feel free to suggest other problems with the argument if you wish, but whatever else is wrong with it, it's certainly not a God of the gaps argument. And that's because we actually do make a positive case, a positive argument. And so try to find a flaw in that argument, sure, but the very fact that we actually make a positive argument means it can't be a God of the gaps argument. There are certain mysteries in science that we don't understand, and yet they don't support theism or atheism. They're equally probable on both hypotheses. So an example of that would be the wave-particle duality of photons, for example. Very, very mysterious and surprising observation. But I wouldn't argue that it supports theism because I don't think there's any reason to think that that's more likely on theism than it would have been on atheism. But with these, with the information content of the cell, the genetic code and its optimization, and the irreducibly complex nanomachines and so forth, I think that these observations are not hugely surprising if we suppose that mind was involved, because we know that minds produce information content, digitally encoded information in particular, information processing systems. We see many of the features of human technology and programming appearing in the cell. We see we know that intelligent agents can visualize complex endpoints and bring together everything needed to realize those endpoints. And so it's not particularly surprising, if we suppose a mind was involved, that you'd make these observations in the cell in biology, but it's wildly surprising on the falsity of that hypothesis. Another way of expressing the argument would be to make the argument from conscious agents, kind of a Richard Swinburnian style, which is to say that on the supposition of theism, it's not particularly implausible that God would create other conscious, in particular embodied agents, because a community of moral agents that are able to interact with others and mold and shape their character in morally significant ways is something that the God of classical theism might plausibly bring about. And it's being embodied, a world of pushes and pulls that accentuates the ability to engage in moral decision making, growing our character, and so forth. And so that sort of world is not particularly surprising on theism, but given all the preconditions that are necessary to get to that world, it's extremely improbable on atheism. And so all of these different lines of evidence that we've looked at that are necessary preconditions to get to the world of conscious embodied agents on atheism could be seen as a part of a cumulative case that supports a theistic understanding over an atheistic one.
Awesome. Yeah, definitely. So no, good answer there. And I appreciate you bringing up Swinburne. That was actually when somebody asked for recommended books. A couple of easy ones by Swinburne are Is There a God and Was Jesus God? That I think would also supplement some of the ones that we've also recommended there. And so Nidemos, I hope I'm saying your name correctly. If God designed genetic code, did he design public lice, cancer, and leeches? So here
Pubic
is
lice.
kind of that. Yeah. Oh, pubic lice. Yikes. Yeah. Well, anyway. Yeah. What about suboptimal design? I mean, we have cases that like congenital diseases, genetic disorders. You have things like Huntington's disease or sickle cell anemia and different things like that. And so you do hear that objection of kind of like evil looking design. So how do you handle that kind of objection? I know that's something that's been brought up since probably David Hume, if not sooner. Very common objection to design.
Yeah, absolutely. I think so. This is not so much the suboptimal design argument as it is the malevolent design argument, right? That basically argues that these systems are designed to cause harm, and so how is this consistent with how is this consistent with the idea of an all-loving, omnibenevolent deity? I don't think it's so much an argument against the design conclusion as it is an argument against theism, and it basically boils down to the problem of evil. Why are there bad things in this world? And I don't think that we have a fully satisfactory answer to that question, and I think that we ought to be candid about that. I think that we ought to be honest and candid when it comes to the full set of evidence that we have, and we understand that there's some evidence that there's a lot of evidence, I think, which is very consistent with and supports and is corroborative of the design thesis and indeed Christianity. And then there's other evidence that seems to be anomalous, and we don't really know how to make sense of it. And I would say that this is an example of that sort of thing. I mean, not to say that there's nothing we can say, but I think this is more of a theological topic than it is a scientific one. And one hypothesis, I'm not saying this is the be all and end all, and I'm also not saying that this solves everything. I don't think it necessarily does. But one hypothesis that I think is interesting is that of William Dembski in his book, The End of Christianity. Is that book you've read, Eric? Or
I've heard of it. I haven't actually read it.
No. Yeah. So basically, Dembski's thesis is that all suffering, including animal suffering, can be attributed to the fall. And traditionally, that sort of perspective has been associated with young Earth creationism. And of course, I reject young Earth creationism. I think it has lots of scientific problems. But what Dembski proposes, and I think this is not implausible, is that the fall is retroactive with respect to its cause and effect relationship, just as the cross is retroactive with respect to its cause and effect relationship, not just benefiting those who will be saved through the cross on this side of the cross, but also those who lived before the time of Christ as well. So likewise, according to Dembski, the fall affects not only creation on this side of the fall, but also retroactively as well, because God exists outside of time. And so on Dembski's model, he sees two worlds intersecting at the Garden of Eden. You have the world that God intended, the idealized world. And then we understand from Scripture it's a local garden. We know this because Adam is placed into the Garden of Eden. And then he's banished, of course, from the Garden of Eden. So it's a local garden. And so according to Dembski's model, so the Garden of Eden represents the idealized world, the world as God intended, where there's no predation or suffering, and Adam would eat from the tree of life, which would mean that he would live forever. But outside the Garden of Eden, you have the world that God has prepared in anticipation for the fall. And so when Adam and Eve fall, they are expelled into that world that God has prepared in anticipation for the fall. And so that's one model that attempts to give an account of that. As I said, I don't think it solves everything, but I think that it is an interesting model. Yeah.
Sure. Yeah, no, it's definitely been one on my reading list. I kind of thought the retroactive thing sounds a little bit strange, but at the same time, you had mentioned kind of the parallel with the cross and how there's that. So I'll definitely have to check that out. Something kind of related to that particular question that I wanted to ask you is cases of like suboptimal design. A lot of times people will bring up things like the structure of the human eye, and they'll say things like the retina is inside out, and the nerves and blood vessels lie on the surface of the retina instead of behind it, as in the case of many introvertebrate species. And this causes, things like blind spots in mammals. And, there's all kinds of other things like, why do I have an appendix when it could rupture? And it's not necessarily, it's like a vestigial organ. Although I think some people have kind of pushed back and said that, well, that helps with like your immunity and different things like that. But, wisdom teeth, all of that kind of stuff. And so, what do you say to people who make that argument of suboptimal design?
The appendix, first of all, is certainly not a vestigial organ, as you yourself mentioned.
I think you're muted. Can you hear me, Jonathan?
I can hear you. I didn't mute myself. But,
it could be me. Hang on. All right, say something. There you are. It was me. My earbuds died. All right.
Can you hear me now?
Yep, I can.
Okay,
go ahead.
Excellent.
Yeah, sorry about that.
No problem. So, all right. So the appendix is certainly not a vestigial organ, as you yourself mentioned. It serves an immunological purpose. It's a reservoir of friendly bacteria. The inverse wiring of the vertebrate retina has also been refuted for a long time now. That argument against design. So the bottom line is essentially this. So retinal cells require a very large blood supply, and because they require a large oxygen supply, so they need to have a very large blood supply as a consequence. And blood cells, of course, absorb light. And so if blood cells invade retinal cells, then the result of that can be blindness. And so that means that the retinal cells have to receive a blood supply from vessels which don't block the light. And so since red blood cells readily absorb light, that demand requires that the retina be wired in the manner in which it is. Now, it's often pointed out, of course, that squids and octopuses have correctly wired retinas that face outwards. And that's completely correct. But these organisms are exothermic, right? They don't require the same blood supply to the retina. So that argument also, I don't think is particularly strong. I would argue that this whole argument from suboptimal design is really an inverse, if you will, of the God of the gaps fallacy, except it's not a God of the gaps fallacy. It's a Darwin or a naturalism of the gaps fallacy. We can't explain why a designer might have done it that way, so we'll just plug in Darwin as a placeholder for that of which we are ignorant. And and as science progresses and advances and discovers why things are designed the way they are, that argument has to retreat with each new advance in scientific discovery. And so it's difficult to say this system, a designer wouldn't have designed it this way, and then we discover later actually a designer would have designed it this way, and there's a good reason why it's designed this way. So I think that argument is not particularly strong. But even supposing that it were the case that there are systems that are designed suboptimally, well, even then, I would argue that even supposing that those systems, these features, are quite improbable on the hypothesis of design, they're still much more probable on the hypothesis of design than they would have been otherwise. And so it still, I think, confirms the design thesis. And there could be developmental constraints, and if one subscribes to the proposition of common descent, there could be evolutionary constraints which make it built the way it is, and so forth. So I don't find those arguments to be very compelling.
Okay. Awesome. Nope. Thank you. Sorry about that. Apparently the Google Pixel Buds only have about 90 minutes before they die, even on 100%. So, yeah. Anyway, so couple more things here and then we'll wrap up. Intelligent design is a religious idea, not a scientific idea. So it gets checked at the door. I guess this was in response to saying, do you see it gathering or gaining more credence among scientists? And so what do you say to people that say, well, this is just religion, it's not science?
Well, okay, it's religion. Let's go back to the question of whether it's true or not. I mean, it doesn't matter how we categorize it. I say that in tongue in cheek. I would say I do think that it's a scientific proposition. But I mean, it's based on standard scientific principles and methodologies. And why is it not a scientific idea? And religious ideas tend to involve worship, whereas the intelligent design is religious ideas tend to involve like ritual and worship and ceremony and so forth. Intelligent design doesn't involve any of those things. So it's an argument, it's an inference. It involves the minimal commitment to intelligent causality, and then different intelligent design proponents subscribe to different religions. I'd say probably the majority of them are Christian, but there are people in other religious traditions as well, including Jewish and Muslim scientists. There are even agnostic scientists. There are people who are theists but non-religious. Michael Denton would be an example of that. David Berlinski is another example of an agnostic with theistic leanings who subscribes to intelligent design. There was a book published some years ago by a guy who was at the time at the University of Colorado Boulder called Bradley Monton, who's an atheist philosopher, and his subtitle of the book was, I think it was called Finding God in Science. The subtitle was An Atheist Defends Intelligent Design. Thomas Nagel, who's an atheist philosopher, has also come out as being sympathetic to design. So I don't think that intelligent design is a religious idea, though it has religious implications, I think. And by the same token, of course, one could argue that Darwinism is a religious idea because it has anti-theological implications. And so if we're just talking, if you want to play the implications card, well, okay, then Darwinism also has implications for religion. So why not call it a religious idea?
Yeah, absolutely. It seems like it's just kind of trying to poison the well a little bit there. So
the Big Bang a religious idea because the Big Bang supports theism as well.
Right, exactly. So a couple more here, and then we'll wrap up. Truth Seeker asks, can you explain what's wrong with Ken Miller's co-option objection to irreducible complexity?
With joy. Yeah, this has been refuted many, many times. And unfortunately, there are some people on the internet who never got the memo. First of all, so the one example of so-called co-option, or something that's called exaptation, that Ken Miller put forward was in respect to the bacterial flagellum. And he pointed out that if one removes, the flagellum associated with, Salmonella or E. coli, there's something like 40 different protein components associated with the flagellum. If you remove 30 of them and you are left with 10, then you have the type 3 export system or the type 3 secretion system, which in some organisms can function independently of the flagellum as a syringe that injects poisons into multicellular organisms. So Yersinia pestis would be one example. Yersinia pestis is the causative agent of bubonic plague. And the type 3 export system in the context of the flagellum plays a role in flagellar assembly. So the proteins, as they're making the flagellum, are exported through the type 3 secretion system, through that syringe. And one thing that's been pointed out is that the type 3 secretion system actually appears to have arisen after the flagellum. It's not a precursor to the flagellum. And so this is a significant problem for that particular scenario, because the evidence suggests, I think quite strongly, that the type 3 secretion system arose later as an evolutionary degradation product of the flagellum, not the other way around. And there's a number of lines of evidence for that. One is, first of all, it makes sense in evolutionary terms because there's an evolutionary pressure to be able to swim long before there were multicellular organisms, animals that you would inject poisons into as a bacterial cell. So it makes sense from that perspective. And then also in organisms that have type 3 secretion systems but no flagella, like Yersinia, for instance, they possess the genes for making flagella, even if no flagella are assembled. And so that, again, suggests that the type 3 secretion system came later. There's also the point that the type 3 secretion systems are taxonomically more restricted than flagella. Flagella are much more widely distributed across bacteria, and so that also suggests that the flagellum is ancestral. And there's other arguments as well. There's phylogenetic evidence that indicates this to be the case. There's also the fact that it doesn't really give an adequate account of the assembly apparatus for making flagella. So flagella, unlike man-made motors, actually have an apparatus for assembling themselves, and they build themselves from the inside out. And they have there's a transcriptional hierarchy or gene hierarchy that ensures a timely expression of different components of the flagellum, so that it makes no sense, for example, to start building the propeller before you finish constructing the hook basal body apparatus to anchor it to the cell membrane or cell membranes. So that's so yeah, these are just some of the problems with that. It also it doesn't, to point out that the type 3 secretion system can serve a standalone function and is homologous to the flagellum doesn't give you an account of how it came to be incorporated into the flagellum. In any case, how it came to be incorporated. I mean, these components have to be well matched in order to work together in unison to achieve a higher level objective. And you can't just, it's going to require multiple codependent mutations, I think, to build a flagellum. You can't just add a type 3 secretion system and then suddenly just add all the other components to make a flagellum, to make a functional flagellum. Fly-D, for example, is the filament capping protein in the flagellum, because the flagellum monomers that form the propeller, because the channels through which they have to pass is very, very narrow. It's only 2 nanometers in diameter. The proteins actually have to be unfolded for insertion into the channel and then refolded at the distal growing end. And there's a capping protein called Fly-D that is a pentagonal disc with 5 leg-like extensions that has to turn, and it basically folds the proteins as they come up to the distal growing end. And if you knock out Fly-D, you lose the flagellin subunits into the medium. That's just one example of what happens if you lose one of the components. There's also a protein called Flag-J, which punctures a hole in the membrane, the outer membrane, to allow rod penetration. It has a hydrolyzing activity, basically hydrolyzes the hole through the outer membrane to allow the flagellum to continue growing. I mean, there's just it's just beyond comprehension how it could come about by stepwise trial and error type of process.
Awesome. Okay. Last question here, and then we'll wrap up. Has any Grays 174 asks, has anybody affirmed ID but given a naturalistic explanation to something like aliens did it, or maybe less crude and plausibly defined?
Francis Crick hypothesized what he called directed panspermia, which is the idea that life on Earth was seeded by extraterrestrials, which is, I guess, a way of saying aliens did it. But I mean, that idea is of course few and far between. There are not many people that would advocate such an idea, and the intrinsic plausibility would be astronomically small. I mean, the probability, I mean, it just moves the problem back a level, because then where did the aliens come from? Yeah,
right.
And I don't think that question applies to a theistic God in the same way, because God is transcendent of time and is eternal. And I think that a theistic being is the best candidate for a non-moving mover, for a being which is the ultimate ground of all that is, and exists by necessity of its own nature. So I don't think that the who designed the designer objection applies to God in the same way it would a contingent being like an extraterrestrial alien. And the universe is so vast that it's very unlikely, even if aliens did exist elsewhere, and it's unlikely that even if aliens did exist, it's unlikely to be sentient, civilized aliens. But supposing that they did by some fluke exist, is very unlikely to ever make contact with the Earth. It is huge, the vastness of the cosmos. I mean, from the Earth to the Sun, you're talking about 93 million miles. To the next nearest star, which is Alpha Centauri, you're talking about 26 trillion miles. And then there are about 200 billion stars in our galaxy and about 200 billion galaxies in our universe, in the observable universe, that is. And so, I mean, I just don't think that even if aliens did exist elsewhere in the universe, it's vanishingly unlikely they would ever make contact in any sort of way with the Earth.
Right, exactly. I think I had like a blog post from a long time ago where sometimes people will say something like absolutely ridiculous, like, well, maybe aliens rose Jesus from the dead. And it's kind of like, yeah, that would be one heck of a trip. And it's like not going to happen. It's not really possible. And so let alone why they would want to raise a Jewish person from the dead. And then obviously with this, I mean, that seeding a world, I guess you could see a reason for that more than raising somebody from the dead. But yeah, it's just not plausible at all. And so I think if that's like what you have to resort to, then at that point, I think you're just really desperate to avoid a theistic kind of conclusion. And so, well, awesome. Jonathan, do you got anything else? I think we're going to wrap up, but this was really great. And just a lot of things that are new to me. It might be new to some of the people watching out there. And so, yeah, thank you so much for jumping on and going through this information with us.
Yeah, absolutely. Thanks so much for having me on. And if you're a Christian struggling with doubts about your faith, please feel free to reach out at talkaboutdoubts.com. We would love to have a chat with you about the evidences for Christianity and address any concerns or objections that you have.
Yep. And again, Team of Scholars includes even people, I believe, that are involved in the intelligent design movement. I've seen Casey Luskin on there. I've seen, I believe, Stephen Meyer. And so I know Tim McGrew has written papers on intelligent design, and he's one of the counselors. And so just an amazing assembly of scholars, a tremendous resource that I can't recommend enough. And so, again, that's talkaboutdoubts.com. And again, thank you so much for joining us. And you guys have a great rest of your night.